Welcome. Today, we're going to take a really deep dive into the very architecture of our brain. Not the solid parts, but the hidden, fluid-filled spaces inside. We're going to explore the ventricles and that amazing liquid that flows through them, the cerebrospinal fluid. You can think of this as a masterclass, really, on the brain's internal plumbing and life support system. We'll cover everything from how it first forms in an embryo to the huge role it plays in keeping us healthy. So let's get started. So, let's kick things off with the most basic question of all. What exactly are the ventricles? We tend to think of the brain as this solid, dense organ, but that's really only half the story. Deep inside, it actually has this complex network of cavities, and understanding these spaces is the absolute first step to understanding how the brain really functions. That's right, the brain is partially hollow. These internal cavities are what we call the ventricles, and they're filled with this clear, protein-rich liquid called cerebrospinal fluid, or CSF for short. And this fluid is absolutely vital for the brain. Now, the most important thing to get your head around right from the start is that these ventricles aren't just isolated little pools. Nope. They form a single, continuous, interconnected system. Just to get our bearings, the two big ones, the lateral ventricles, they're located within the cerebral hemispheres, the part of the brain that develops from a region called the prosencephalon, or forebrain. Then there's the third ventricle, which sits deep in the core in an area called the diencephalon, which houses critical structures like the thalamus and hypothalamus. Okay, so here's the roadmap for our session today. We're going to start at the very beginning with the developmental origins of this whole system. From there, we'll jump into a detailed tour of its adult anatomy. Then we'll zoom right in on the fluid itself, the CSF, and follow its incredible journey through the circulation path. After that, we'll look at pathology, what happens when things go wrong, and finally, we'll see how doctors can use the system for diagnosis, giving them a window into the brain's health. All right, Section 1, Origins. To really, truly understand the complex, winding shape of the adult ventricular system, you have to go back in time to how it all began. Believe me, its final, intricate structure makes perfect sense once you see its surprisingly simple start. And it all starts right here, with this structure, the neural tube. As you can see in this embryonic cross-section, what begins as a flat sheet of cells, the neural plate, starts to fold in on itself, making a groove. Then it literally zips itself up to form this completely hollow tube. This simple little structure, this is the precursor to the entire central nervous system, the brain, the spinal cord, the whole thing. And that hollow space right there in the middle, that is the very beginning of the entire ventricular system. Of course, this tube doesn't just stay a simple tube for long. At the cranial end, that's the end that's going to become the head, it goes through these really dramatic expansions. It's like it balloons out in specific places, and these expansions create the three primary brain vesicles, the forebrain, the midbrain, and the hindbrain. The rest of the tube stays narrow, and that's what will eventually become the spinal cord. Now this table right here, this is the blueprint. This is what connects that early development to the adult anatomy we see. Let's walk through it because it's so important. Look at that first row. The primary vesicle is the forebrain, or prosencephalon. It then divides into two parts. The first is the telencephalon, which just blossoms into the big cerebral hemispheres, and its internal cavity expands to become those two large lateral ventricles. The second part is the diencephalon, which forms the thalamus and hypothalamus, and its cavity becomes the third ventricle. Moving on down, the midbrain, or mesencephalon, it doesn't subdivide. It just develops into the adult midbrain, and its tiny little internal channel becomes the cerebral aqueduct, a critical connection point we'll talk more about in a bit. And finally, you have the hindbrain, or rhombencephalon. It splits to form the cerebellum, pons, and medulla, and its cavity expands into the fourth ventricle. And right at the bottom, what's left of the neural tube becomes the spinal cord with its tiny central canal. So what this table shows us so perfectly is that every single major part of the adult brain has a corresponding internal space that grew right along with it. Okay, so now that we've seen where they come from, let's take a proper anatomical tour of the fully formed system. We're going to map out these internal cavities exactly as they exist in the adult brain. We'll start with the biggest players in the system, the two lateral ventricles. As you can see in this image, there's one in each cerebral hemisphere. And you can't miss their distinctive C shape, which kind of sweeps through all the different lobes of the brain. These are the big cavities that developed from that telencephalon we just talked about. Let's get a bit more specific about their boundaries. Each lateral ventricle has a central body, and then these extensions, which we call horns, that reach out into the brain's lobes. There's an anterior horn poking into the frontal lobe, a posterior horn going back into the occipital lobe, and an inferior horn that curves down into the temporal lobe. So what's around it? Well, its roof is formed by the corpus callosum, that huge bundle of nerve fibers connecting the two hemispheres. Its floor is made up of two deep brain structures, the caudate nucleus and the thalamus. And medially, separating the two ventricles, is this thin little membrane called the septum pellucidum. Now, this is a really crucial point. The two lateral ventricles do not communicate directly with each other. They're neighbors, but they don't have a door between them. So, if they don't connect directly, how does the fluid move on? Well, the exit route from each lateral ventricle is this little channel called the interventricular foramen, also known as the foramen of Monro. It might be small, but it's absolutely vital. If you want to know its exact location, it's bounded at the front by a structure called the fornix and at the back by the anterior end of the thalamus. All right, so if we follow the flow of CSF through that foramen of Monro, we arrive at the third ventricle. This is a very narrow, kind of slit-like space right on the brain's midline, basically sandwiched between the left and right thalamus and hypothalamus. From here, the CSF flows into another critical, narrow channel, the cerebral aqueduct, or the aqueduct of Sylvius. This little passage connects the third ventricle down to the fourth ventricle. The fourth ventricle has this really distinctive tent-like shape. It's situated in the brainstem, nestled between the pons and medulla in the front and the cerebellum in the back. Now, at its bottom, it tapers down to become the central canal of the spinal cord. And this is super important, it also has 3 small openings in its roof. And these are the exit doors from the entire internal ventricular system. This diagram here really puts the whole connected system into view. Let's focus on that final exit path. CSF gets out of the fourth ventricle through those 3 little openings we just mentioned. There are 2 on the sides, called the foramen of Luschka, and a single one in the middle at the back, called the foramen of Magendie. These 3 foramina are the crucial gateways that allow the CSF to finally escape the internal ventricular system and flow into what's called the subarachnoid space, which is the fluid-filled layer that surrounds the entire brain and spinal cord. So we've just toured the containers, the ventricles themselves. Now it's time to shift our focus to what's inside them. This whole section is all about the fluid itself, the cerebrospinal fluid, or CSF. So what exactly is this stuff? Well, when it's healthy, CSF is a completely colorless fluid. Chemically, its mix of inorganic salts is actually very similar to what you'd find in blood plasma. It also has glucose to feed the brain cells, really small traces of protein, and a few immune cells called lymphocytes floating around. And this whole system is kept under a constant, gentle pressure, usually somewhere between 8 and 15 millimeters of mercury. And how much of this fluid do we have in our heads at any one time? Well, you might be surprised. It's a pretty small amount. Just 135 to 150 milliliters in an adult. To put that in perspective, that's about the same as a small cup of coffee. But the job it does is absolutely massive. And to understand just how massive its job is, look at this comparison. An adult brain, if you were to take it out and weigh it in the air, is about 1,400 grams, around 3 pounds. But when it's floating in cerebrospinal fluid, its effective weight is reduced to just 50 grams. That is incredible buoyancy. That is what stops the brain's own weight from crushing all the delicate blood vessels and cranial nerves at its base. It's literally floating. So let's quickly summarize the 4 key functions of CSF. First, there's protection. It's a liquid cushion, right? It absorbs shock and buffers the brain from getting banged around, like in a concussion. Second, buoyancy. Like we just saw, it dramatically reduces the brain's effective weight. Third is excretion. The constant one-way flow of CSF is like a garbage collection system, carrying away waste products, toxins, and drugs from the brain into the blood so they can be disposed of. And fourth, it acts as an endocrine medium, carrying hormones that are made in one part of the brain to other, more distant parts of the brain. So where does this amazing fluid actually come from? Well, the main factory for CSF is a specialized tissue called the choroid plexus. And this diagram here shows you its unique structure. You've got a core of leaky blood capillaries, and they're surrounded by a layer of special epithelial cells called ependymal cells. And see how these structures are all folded up, like little fronds? That's to maximize their surface area so they can produce a lot of fluid. The vast majority of CSF is made in these choroid plexuses, which you find in the lateral, third, and fourth ventricles. A little bit extra is also thought to be secreted by the ependymal cells that line the entire system. But now, get ready for a really stunning fact. The production rate is about 500 milliliters every single day. Now remember, the total volume in the whole system is only about 150 milliliters. That means the entire volume of CSF is produced, circulated, and then replaced about 4 times a day. This constant recycling is so important for its cleaning function, flushing out all that metabolic waste like the beta-amyloid proteins that are involved in Alzheimer's disease. And after it's done its job, its final destination is to be drained back into the blood. This idea of constant production and drainage leads us perfectly into our next section, the complete journey of CSF circulation. What we're going to do now is follow a single droplet of CSF from the moment it's created to the moment it gets reabsorbed, tracing its entire path of the central nervous system. It's quite a trip. Okay, let's trace the path right here on this diagram. First, the CSF is made in the choroid plexuses of the lateral ventricles. Then, it flows through that interventricular foramen of Monro into the single third ventricle. From there, it passes down through that narrow cerebral aqueduct into the fourth ventricle. Next, it exits the internal system completely through the foramina of Luschka and Magendie, and it enters the subarachnoid space. Once it's out, it collects in these large pools called cisterns, like the really big cisterna magna at the base of the brain. And from there, it begins its slow journey, circulating up and over the entire surface of the brain and down around the spinal cord. And this isn't just a passive float. It's actively helped along by the pulsing of arteries, by our breathing, by coughing, and even just by us moving our bodies around. So this long journey has to end somewhere, right? The CSF has to get back into the venous blood. And this critical step happens through these specialized structures called arachnoid villi, or arachnoid granulations. Basically, they're these little mushroom-shaped protrusions of one of the brain's protective layers, the arachnoid mater, that poke right through another tough layer, the dura mater, and into the large venous sinuses that collect blood from the brain. Their entire job is to act as one-way drains for the CSF. And the way they work is just brilliant. It's a completely passive one-way valve system that's driven entirely by pressure. Here's how it works. Step 1, under normal conditions, the CSF pressure is just a little bit higher than the blood pressure in the veins. This pressure difference pushes the villi open, and that allows the CSF to flow from high pressure to low pressure, draining right into the bloodstream. Simple. But if the situation were to flip, if for some reason venous pressure got higher than CSF pressure, that same pressure difference would push the flexible tips of the villi closed. It's this beautiful, simple mechanism that makes sure no blood ever flows backwards into the sterile ventricular system. So we've seen the system in a state of perfect, delicate balance, where production equals absorption. But what happens when that balance gets thrown off? Let's now move into the pathology of the ventricular system and look at what happens when this intricate machinery fails. And this image right here shows the devastating consequence of that failure in the most dramatic way possible. On the left, you've got a cross-section of a normal brain, and you can see the ventricles are just these narrow, dark slits. On the right, that's a brain with hydrocephalus. The ventricles have become massively blown up by trapped fluid, and they're compressing and tragically damaging all the surrounding brain tissue. So what is hydrocephalus? At its core, it's an accumulation of excess CSF in the ventricles, which causes the pressure inside the skull to rise. And this can happen for one of three reasons. First, the choroid plexus is making way too much CSF. This is pretty rare, though. Second, there's a blockage somewhere in the system that's stopping a flow. Or third, there's a problem with those arachnoid villi, and the CSF isn't being absorbed properly. It can be congenital, meaning you're born with it, or it can be acquired later in life, from an injury, a tumor, or an infection. The standard treatment is a surgical decompression. A thin, flexible tube called a shunt is put into a ventricle, and that tube is then tunneled under the skin to drain the extra CSF into another part of the body, like the jugular vein or the abdomen, where it can just be safely absorbed. We can also classify hydrocephalus into two main types, and it all depends on where the problem is. In non-communicating hydrocephalus, there's a physical blockage somewhere inside the ventricular system itself, like a tumor plugging up that narrow cerebral aqueduct. This physically stops the CSF in the ventricles from being able to communicate with the subarachnoid space. Then you have communicating hydrocephalus. Here, there's no internal blockage. The system is wide open. The problem is happening outside the ventricles, usually because of faulty absorption at the arachnoid villi. Now, all this increased pressure inside the skull from something like hydrocephalus has consequences that you can actually see outside the brain itself. To understand one of the most important clinical signs, we need to look at this diagram of the optic nerve. You'll notice that the layers that surround the brain, the meninges, and that CSF-filled subarachnoid space, they don't just stop at the skull. They actually extend all the way along the optic nerve, right to the back of the eyeball. The nerve is basically bathing in CSF its whole length. This unique piece of anatomy leads to a condition called papilledema. Let's break it down. Cause, mechanism, and consequence. The cause is a rise in the CSF pressure inside the head. The mechanism is that this high pressure gets transmitted directly along that sleeve of subarachnoid space around the optic nerve. This pressure then squeezes the thin walls of the retinal vein, making it hard for blood to drain out of the eye. And the consequence? You get a fluid backup, causing the optic disc, the head of the optic nerve, where you see it in the eye, to swell and bulge forward. That is papilledema. And if it's not treated, it can lead to optic atrophy and, tragically, permanent blindness. And all of this brings us to our final section, diagnosis. Because the CSF is in such close contact with the brain and spinal cord, taking a sample of it, usually through a lumbar puncture or a spinal tap, can give doctors a powerful window into the health of the entire central nervous system. So let's run through what different findings in a CSF sample can tell a clinician. If the fluid, which should be crystal clear, looks cloudy, that suggests there's a lot of protein or a lot of white blood cells, which is a big red flag for infection. A yellow color, which we call xanthochromia, is caused by old, broken-down red blood cells, which means there was probably a bleed into the CSF sometime in the past. A high white blood cell count points directly to active inflammation, like you'd see in meningitis or encephalitis. Increased protein levels suggest the blood-brain barrier has become leaky. Finding fresh red blood cells is never normal. It means either the needle caused a little bleeding during the tap, or, much more seriously, there's an active hemorrhage in the brain. And finally, just measuring a high opening pressure during the procedure itself can point to a tumor, an abscess, or swelling from meningitis. When you step back and look at the whole thing, the crucial takeaway is that this entire system is a masterpiece of biological engineering, maintaining this incredibly delicate, dynamic balance of production, flow, and absorption. So having seen its complex anatomy and its vital functions, I want to leave you with this question. What single point of failure in this whole system do you think would cause the most rapid and the most severe neurological damage? It's a question that really drives home just how fragile and how absolutely essential this incredible system truly is.